So my name is Yi. I will be sharing with you our view on the rising impact of RNA-Seq in microbial transcriptomics. So sitting next to me is Megan, our distributing account associate. So both of us will stay the entire course of this discussion session. So we're looking forward to the chat. So in our discussion, we will be discussing the following topics. Now start with some statistics to show the increased use of RNA-Seq in microbial research. And then we'll continue to elaborate Zymo researchers' tools on how we react to such increasing and expanding use of this technology in microbiomics, being that ribo-free is a technology that we believe can be a powerful tool for microbiomic studies. Now, I'll briefly discuss the principles. So how does ribo-free work? And then more importantly, sharing with everyone some data on our internal challenging of the system to low, medium, and high complexity microbial community samples that it will mimic in real research lives how different complicated extents microbiomic samples will look like. And following on will be some use cases that I believe speaks louder because it shows how the community are applying this technology to all kinds of interesting, exciting research that have already been made public in peer-reviewed publications. And in the end, I'd like to reiterate that Zymo research is here and we're ready to help with innovative technologies and stable supply. To work with Techno in advancing technologies in your area for research to continue to advance and enhance humans' knowledge of nature, after all. So without further ado, let's just jump to why we think that there is an increasing use of RNA-Seq in microbial studies. So looking at the y-axis here, and let me see if I can make it a pointer. And indeed I can. So on the y-axis here is the number of publications. On the x-axis here, you're looking at individual years covering the past 10 years. Now we're seeing an increasing number of microbial transcriptomics papers being published over the past 10 years. Exemplified by this increasing blue bar. And the same upward trend actually is seen in this orangish bar, where we specify RNA-Seq being the method paired with microbial. And alternatively, when we focus on a specific subcategory or application within microbial studies, that is meta-transcriptomics, we're looking at the same upward trend represented by the increasing green bars here. We're just in half of March this year in 2025, and we're already over 1/6 or 16% of last year's. So looks like in 2025, we'll have the equally exciting trend of seeing more work coming out. So all of this, from my perspective, demonstrates the fact that we're looking at expanding popularity of NGS-based RNA analyses in microbiomics. So how does Zymo research respond in this trend? We provide premium NGS technologies and offering. And this is actually an emergent section here at Zymo research. So for some of you might already know Zymo from our legacy offering, as showcased here by this rainbow of offering. We provide sample collection technologies to nucleic acid extraction and sequencing services with a dedicated microbiomics team. And after all, we are known as the epigenetics company. Of course, we have epigenetics section, products and services. Now, we also have, for instance, my team, RNA analysis group, along with our sibling team in epigenetics and microbiomics, that we're providing Zymo-Seq, this family of products, to provide for the needs of NGS solutions, among which our ribo-free RNA-Seq is a flagship offering that we're very proudly to introduce to all. So why we think ribo-free or ribosomal RNA depletion is important for total RNA-seq? The answer actually is very simple, because rRNA really takes up a lot of a very common general RNA content that you can extract from your samples. So in this pie chart, you're looking at the majority being this blue, representing rRNA. And from what we see, either in our own experience as well as from publications, that rRNA can make up to 90% or even more sometimes of your RNA sample. This means that in many research applications, a lot of the information that you get from your sequencing libraries actually are not informative for the questions that you're looking an answer for. But because rRNA are so many that they take up the majority, they will consume most of the sequencing reads, which then significantly reduce the efficiency of any sequencing run. And therefore, naturally, scientists would want rRNA depletion methods to reduce the signal coming from rRNA and in turn increase their experimental efficiency. But what we've seen is that conventional methods, including those on the market now, require organism-specific probes. This means for scientists who are studying less common or non-model organisms, such as not human mouse rat, which includes microbes, may require custom probes. What this means is that this procedure is quite expensive, because not only you need to design, test, and modify, but it may also not work. And even luckily, it eventually works. The whole process is very time-consuming, because you also have a lot of bench work that is very labor-intensive before you have a tool to utilize and answer the biological questions that you are originally interested in. Therefore, Zymo's solution is to provide a tool called ribo-free, where it has this unique feature that I think, as of now, we are still the only provider that has universal RNA depletion for any organism. So how does ribo-free achieve this? The quote-unquote secret actually relies on RNA themselves. They are so overly abundant, they will drive the depletion of the cDNA coming from them. So after reverse transcription or cDNA synthesis, we will denature the DNA-RNA hybrids and try to renature. During this process, because rRNA are so overly abundant, paired with the cDNA coming from them, these two are much more easily pushing this equilibrium to the right side for the duplex to form. And once the duplex forms, enzymatic digestion kicks in to remove the cDNA from the rRNA, but leaving the cDNA from other types of RNA molecules remain in the reaction that's ready for the subsequent steps in the library prep procedure. And as you can see in the whole process, there are no probes being part of the processing, which means we virtually lifted that species limit. There's no limit on which one species the RNA can come from to be applied to this ribo-free procedure. In other words, ribo-free makes a great tool to expand whole transcriptome profiling beyond human mouse rat. Now, and of course, ribo-free works with human mouse rat very well. And here we're looking at our validation data of ribo-free RNA-seq on various organisms. And the sequencing efficiency is significantly improved across all of them. The green bar represents protein coding and other RNA signals. And this orange bar represents rRNA. So simply put, the less or the shorter the orange bar, the merrier. And looking at these libraries prepared from samples across a wide variety of biological kingdom of phyla, including human, rodents, cow, avian, and plants, insects, and of course, microbes. So for all of these different species, ribofree RNA-Seq was able to reduce the orange bar to be below 80%, and in many, rephrase, ribofree RNA-Seq technologies were able to reduce the orange bar, which is all RNA, to be below 20%, and in many cases below 10 or even 5%. So in other words, your informative or quality reads are more than 80%, a lot of time over 90%. Now, those data are applying ribofree to its canonical use, that is single species, for very regular RNA-seq analyses. Looking at its mechanism, one thing that is actually coming up is that when we have samples coming from mixed species, which is very common in microbial community samples, the RNA depletion efficiency might be reduced. Is that true? And if true, how much are we seeing it reduced? We started validating ribofree to broaden its application into meta-transcriptomics samples, not just one single strain, as we see earlier in the yeast data. So we first tried with our microbial community standards. That is a sample that has 10 strains of bacteria and fungi all mixed together. And the results is actually very encouraging and decent. It showcased that this technology still increases efficiency, even in mixed, challenging microbial community samples. Looking here, we have libraries that are prepared with ribofree, and we also emit the ribofree depletion during library prep as a control group. The all RNA reads percentage, if we don't treat it with ribofree, it's close to 98%. So almost all of the reads go to the not as informative portion. While if we look at the treated one, that we significantly reduced the reads to be below 40%. And this means we significantly enriched quality reads or informative reads from single-digit percentage all the way to over 60%. This is a 26-fold increase. With all of these increase in quality reads in low to medium complexity samples, such as our community standards, researchers will have more information for higher quality subsequent analyses. And such as what we tried here. We utilize these information. And remember, ribofree RNA libraries are short read or NGS libraries. They using these info to do reference-free de novo transcriptome assembly will be slightly harder than using, for instance, long reads signal. But even under this very unfavored conditions, our library will still be able to double the maximum contig length in ribofree libraries comparing to not treated one. And this doubling increase or the increased efficiency also got reflected in N50, where that simply put, the larger the value, the better in the data quality. So to briefly summarize is that with community samples, low to medium complexity, ribofree technology improved efficiency, double-digit fold increase on the quality reads. And even if you apply the data to very challenging analyses, thinking these are short read data, we're still able to improve a lot on the contig length that one can get through de novo assembly. And we didn't stop there. And we further asked a question. Okay, if using a low to medium complexity community sample, we got very good data, how about even more challenging samples, such as gut and fecal reference? So these are medium and high complexity samples that are in general very challenging for any methods to handle from my perspective. And the data is still very encouraging. And we were very happy when we seen that, because we know that this proves ribofree being a powerful tool for almost for most aspects in the microbiomic research as of now. So showing here in the green background data are the gut standard, and we tested with 250 nanogram of RNA as input, as well as 100. At 250, our non-RNA reads, in other words, quality reads for any subsequent analyses, are 60%. And remember, had we not treat these RNA with rebel-free library prep, there likely would be over 90% being RNA reads. In other words, will be below 10% or even below 5% being these quality reads. And similarly, for the fecal reference, we also are reaching close to 50% when we use 250, the maximum input our kit allows, or still very good, close to 40% of useful reads. That is more than 1/3 of your library raw reads, even when you use 100 nanogram of RNA. That is in the middle ground of our input range. We still have over 10 kb, so we are on the double-digit kb contig length using a short read library data to do reference-free de novo assembly. And all of these results showcase that for challenging medium to high complexity mixed microbial samples, the rebel-free technology, even not designed for this purpose, are still helpful in enriching for informative reads and enhancing sequencing efficiency for downstream metatranscriptomics analyses. Now, that's our internal data invalidation. But to a certain perspective, what's more encouraging is when I look at how the scientific community are applying this technology. So 2 days ago, I was looking on Google Scholar, and to my surprise, in a good way, even though our rebel-free technology is so young, it's only on the market for a few years, we already garnered over 250 peer-reviewed publications so far. And when I look at the field of applications, it was so exciting to see that probably only the imagination is the limit for scientists to explore. I found samples from all of these publications come from human, mouse, rat, which are not surprising because they're likely the most studied species still. But I've seen other mammals, worms, fish, insects, plants, and of course, bacteria and fungi in those studies that have been published. And then the research topics also ranged widely. We have, of course, the regular use case for RNA-Seq, that is to do gene expression profiling. You're quantifying the transcriptome levels. But I also see the functional annotation of genomes. I've saw studies in the field of plant molecular biology. Not to mention agricultural virology is also on the rising side. And of course, a significant portion of the publications are coming from microbial transcriptomics, among many other applications. So let's get to the third part of this discussion, that is to showcase a couple of interesting papers that I've seen that how scientists are utilizing rebel-free in various research and applications. So the first three will be more general, are beyond microbial transcriptomics. And then I'll share three that's focusing on different aspects of microbial research. The first one is about chicken. So this group of scientists coming from University of Washington and Western University of Health Sciences utilized rebel-free RNA-Seq to do functional annotation of farm animal genomes. Chicken is probably the #1 consumed agricultural avian species. And specifically, this group of scientists successfully characterized 10 cell and tissue types of their transcriptomes. They were able to identify protein coding transcripts and long non-coding RNA transcripts that are specific to each of these cell and tissue types. And then the next example come from insects, Drosophila, that the group from Yale utilized rebel-free RNA-Seq data to profile long non-coding RNA landscaping. Their models were Drosophila and 2 different strains of flies for neuroscience. a sneak peek of their main discoveries is that this group found hundreds of linear long non-coding RNAs present in Drosophila antenna. And among these, they found that 74 were previously unannotated. This means that this group of researchers successfully identified novel linear long non-coding RNAs in their study. Now, the third example, getting closer to microbial transcriptomics, that is plant virology. So this group of researchers utilized BENZ as the subject of study, and they successfully identified differentially expressed genes of this host, BENZ, when it reacted to the infections of two strains of the red clover necrotic mosaic virus. One is a wild strain, the other is a specific mutated strain. So knowing more information of host mechanism reacting to the virus is always a very good knowledge gaining step for human to have a better idea on later the diagnosis and prognosis of the plants once they get after they get this virus infection. So these are the three examples of how researchers have been applying RNA-Seq powered by RABO-free in many different fields. And the next is let's focus on microbial transcriptomics. The case study number 1 is about one single strain. This is MTB, the Mycobacterium tuberculosis, the bacteria that's responsible for TB in human. Specifically, this team that's primarily based in Texas, US, they exposed 4 different TB strains to human alveolar lining fluid for different time durations. And then they performed differential gene expression analyses on the RNA libraries that they sequenced and detected close to 400 differentially expressed genes. They found that these genes are associated with multiple important pathways, such as lipid metabolism, cell envelope and processes, as well as other metabolism and respiration functional pathways. This suggests that the TB's early contact with ALF drives metabolic and physiological changes in this bacteria. So this group of authors believe that this knowledge highlighted the importance of the host's lung environment and its interactions with the bacteria during the infection process. So this is again another example of microbial transcriptomics of bacterial pathogens can add into humans' knowledge of how in the future that we can better battle against these diseases. Okay, so we have a single strain of bacteria as the subject of study. The next case is about viral pathogen. Specifically, this group of researchers were able to assemble viral pathogen genome from a deep archived museum sample. So how did this happen? Briefly, researchers prepared RNA libraries with heart and kidney RNA from a 20-year-old frozen museum sample that's collected from a Costa Rican pygmy rice that was originally collected in Panama. And it was really a long journey for this piece of sample, really precious. And then with these libraries, they deeply sequenced it and successfully assembled a coccovirus genome. And the reason that they are interested in this coccovirus being one of the hantavirus is that hantaviruses can cause severe diseases in human, but they can also maintain in rodent populations without harm. So they believe that this piece of sample could provide very valuable insights. And indeed, in this successfully assembled coccovirus genome, the scientists found that their genome had 91% of complete consensus against the reference. But they also noticed a 1.1% variations that's different from the reference. they further identified 8 non-synonymous mutations. They further did more analyses using their own data coming from ribofree RNA libraries, as well as publicly available coccovirus data, and identified a clear relationship between clinical cases of this type of virus, as well as this specific strain that got acquired in this rice rat that's once collected in Panama. Through this trajectory of study, this research showcased the significance is that for viruses at low abundances, they can still be recovered from deep archived tissues, such as now hosted in museums. And such type of samples would provide valuable genomic data and tools for future surveillance and diagnostics. And at the end, the knowledge will all contribute to humans' better understanding of virus evolution and pathogenicity.
A question, would you like to see it?
Of course. Megan was telling me that there are questions coming in the chat. If we don't mind, I'll address them all at the end. I really appreciate it for your interest and patience.
Yeah, for sure, doctor. No worries.
Okay, so now we have a single strain. We have virus. How about when things involving host? And indeed, among those publications utilizing ribofree, there are such type of use case. And this is about host microbe interactions in those patients with diabetes-related foot ulcers. RNA-Seq libraries were prepared from tissue punch biopsies from 2 groups of patients. Those that have acute infections of DRFU, so diabetes-related foot ulcers, and those that have localized chronic infections. Through analyses, this group of authors were able to identify distinct host microbe interactions between these 2 conditions. And specifically, they found that in those with acute infections, the upregulated genes are more related to vascular inflammation, T-cell signaling, and olfactory receptors. While for those that has local chronic infections, upregulated genes are more about cellular homeostasis maintainment and biofilm formation. And therefore, they drew a temporary summary using information that's made available to them as of now, that bacteria in acute infections of diabetes-related foot ulcers would prioritize their motility and pathogenicity, while bacteria in chronic infections are more focusing on biofilm-associated genes, trying to stay the homeostasis that they are in. So with all the information that I've shared with all so far, hopefully that I've conveyed this message that ribofree RNA-Seq is a powerful tool to microbiomics. It can be used in single strain, which is its canonical designed purpose, but it is also helpful in improving efficiency for research with mixed species, including host microbe samples, where you both have the human host or other mammal host along with microbes. Furthermore, use cases for viral pathogen detections has been shared, showcasing its wide adaptability and flexibility. So by now, we've focused on ribofree, and we've mostly talked about RNA-Seq. And like to leverage this opportunity actually to bring up this information is that Zymo Research actually provides the technologies that cover the entire RNA-Seq sample prep workflow. From collection, you have DNA RNA shield that can enable room temperature preservation transport. From extraction, we have 3 different lines that can accommodate different types and samples that have specific needs for NGS-grade RNA extraction with high efficiency. And of course, we have RNA-Seq offering that ribofree is a great tool with the simplest RNA library prep with universal RNA depletion for any organism. With these reagent offering, if researchers don't want to perform these bench work by themselves, we have services department that can help scientists carry all of these steps. But regardless if it's research, if it's reagent or services, we have data analysis support. We will help researchers to perform data analysis once you obtain the raw sequencing results. And our goal is to support for reagents as well as scientists using these reagents to have publication-ready style of data visualization and reporting. And finally, last but not least, another callback is that Zymo research actually provides multi-omic solutions, including reagents and services. And we've talked about ribo-free, the one-for-all solution for total RNA-Seq. We also have reagents for 3' mRNA-Seq, that's more for efficient mRNA expression quantification. Our services team also provide microRNA sequencing services. And similarly, on microbiomics side, we have reagents for 16S sequencing and services for shotgun metagenomics. Not to mention that with ribo-free, we're also able to perform metatranscriptomics services. And epigenetics side, we have well-known technologies to study DNA methylation and chromatin accessibility profiling. So with this, I'd like to thank you all for your attention, and we can start our Q&A discussion. And David, of course, is your direct point of contact, but Zymo Research is also here to work with Techno and David to continue supporting you. So I have my email address and my colleague Megan's email address listed here. And if these are not easy for you to memorize, we always have tech support email address readily available for you. So I'll go back to this slide and let's do Q&A. Thank you all.
Thank you, Doctor, for the information that you shared with us. No sé si por acá los asistentes tienen algunas dudas, sino para compartir las que tenemos en el chat. Frank, Nazaret, bueno, bienvenidos, gracias por unirse. Ezequiel, Harold, si algo tienen por ahí, que bueno, se sientan libres de compartirlo. Doctor, we have some questions in the chat from Luis. Luis say, how can these ribo-free libraries can be preserved or storage in case we want to send some libraries abroad? Doctor,
Got it.
there is
Of
some
course.
alternative from Zymo? Or...
Yeah. So if Luis, you're looking at shipping libraries, libraries are double-stranded DNA by the time they become libraries, so they should be fairly stable. Blue ice shipping should be sufficient.
All right.
But if you're thinking about shipping samples, so the RNA directly, then one method is to utilize shield to enable room temperature transport. Another method possibly is using dry ice packaging for shipping abroad.
All right.
Mm-hmm.
There is
I
an
hope that
alternative.
answers.
Sorry, there is an alternative to send samples or libraries with room temperature.
I'm sorry, would you mind repeat your question, David?
If we are able to send samples at room temperature with their ribo-free libraries.
I see. So for libraries, because they are double-stranded, the risk of shipping them at room temperature is small. However,
Right.
I would not recommend doing so because especially if it's overseas or overseas transport, there might be some hiccup happening. There will be unexpected delays. And therefore, a good middle point is to use blue ice shipping.
All right, great.
Yeah. Mm-hmm.
And the other question is if we are able to run these libraries in other technologies more than Illumina, such as MGR, such as MGI or Oxford, Doctor?
Yes. So Rebel-free libraries are next-generation sequencing libraries. And therefore, while it is possible to sequence them on Oxford Nanopore sequencers, the efficiency may not be there, meaning that technically it is totally possible. We actually have customers sequencing Rebel-free libraries on Oxford Nanopore, but just be mindful that these libraries are NGS length, so they are short. They are shorter than traditionally known as long read, which are probably several KBs. And there would be bioinformatic considerations to sequence these libraries on Oxford Nanopore platforms. So if there are interest, we can definitely discuss more details further. Feel free to contact David and Megan or me via email. But the short answer on Oxford Nanopore was that while it's technically possible, there are more detailed considerations if we really want to move forward like so. On the other hand, for MGI, MGI system, the vendor provides conversion kits. And I believe there is a MGI conversion kits that has App A, so application A, App A in their kit name, that will convert Rebel-free libraries from Illumina structure to MGI structure.
All right.
And we have feedback from customers that they successfully sequenced our Rebel-free RNA-Seq on MGI platforms when they follow MGI providers' suggestion in converting in library conversion. So for MGI, the answer is yes, they are compatible. Please use MGI's conversion kit, and then the rest shall be quite straightforward.
All right, great.
And then another add-on point is that it's not mentioned here, but besides Illumina, Rebel-free libraries are also compatible with Element Biosciences VD system. So especially with VD's most advanced flow cell, it is a straight direct loading. The conversion is already part of their flow cell. So in other words, there are no hands-on conversion needed. You can use our kit as they are and load the libraries to a VD's freestyle flow cell per manufacturer suggestion. For a VD's earlier generation of flow cell, similarly to MGI, the vendor, Element Biosciences, provides conversion protocol. That's very simple to implement. So just add one step of conversion, you will be able to sequence Rebel-free on Element VD as well.